your Mac. If you have a Mac, it's an MP4 file, or you have a PC. You can load it on. You can load it on. If you have a problem reading it on your phone or anything else, you can use a program called VLC. VLC is a media player. It plays any kind of media file. So you have no problem. Are you ready? You got your water ready? Good. So let's start with the intro to the course. All right. So I guess we should start this as to why you're here. And that's, I guess, about as good a beginning as I can think of. The reason you're here is for us to study what histology is. It is for us to understand that cells and tissues, which we're going to clearly speak about, are, it didn't download. Have you noticed this? This is kind of interesting. Cells and tissues have a distinct role in how we study materials. It is to understand that histology, that is not your lecture, so don't panic. Okay? So it's to understand what relationships are. And I think part of our problem is that we tend to only want clinical materials, but without an understanding that you don't do clinical without some basic science, because everything does somehow relate. That you can't understand inflammation unless you have a background. You can't understand development of the face unless you understand it comes from tissues. You have to really understand what the concepts are that we deal with. This is your textbook. It is they have, if you buy it and get access to the course, they have lots of materials, but they do have self-evaluating questions. It is not the best book in histology and oral embryology. There are some much better ones. However, their pictures are great. I am a visual learner. I think the pictures here are awesome. The text-based material is okay. It's not the best, but it's good. It's good for the most part. And that is your required textbook. I understand we have a very short period of time. I guess the best book, there's Tenkati's Oral Histology. There's a much later one that just came out. I do not recommend buying it. It's really made for dental students and people who like to torture themselves. And I do like this Fehrenbach. Are you familiar with the author Fehrenbach? No? Fehrenbach, besides writing this illustrated dental embryology, histo, and anatomy, wrote a wonderful book. You may have used one of the PowerPoints set up when you took A&P that goes over the muscles of mastication and what they do. So she's a really great writer. I like the book, but the pictures are it's a little bit more than I like to do. So the first part of the course really depends on the book that you may have sold, and that's your anatomy and physiology book. But I think I have enough material to get you going. So basically, it's really a number of courses, a number of different topics. I do put up grades, kind of interesting. I do not give a cumulative final. I have a history of not doing that. One, for this course, your final material is difficult enough without me adding additional questions on. Passing grade is 75. You know where you are. Very, very simple. Again, I need to get a call from you. You do not want to take my makeup exam, trust me. It is short answer, it is multiple choice, and long essay. Okay? Okay. Please, I understand you all have lives to live, and you all want to text instead of looking at me. But sometimes what drives me crazy in a class is students who are doing the bobblehead thing. They're going like that, and they're looking at their text. Now that you all have computers, you can now search on the internet and have a good time and do whatever you have. So I ask you to please stay on tack. Okay, I do want a middle aisle because I like walking. I don't want to get tired. So I walk around the room so you can hide things. So basically we started with basic histo. basic cell, facial and oral developmental biology, and tooth development, where we start here. Number 2 is really facial and really developmental biology. We start there and we continue it in Unit 2. And then Unit 3, we do overlap in tooth development. And we do really everything about eruption, exfoliation. We do everything about the teeth. So what we're interested in studying is the microscopic structure of bone, whether or not it's stratified squamous non-keratinized tissue, or whether or not we're looking at a tooth. I use a lot of pictures to make it a visual thing, or when we're looking at a tooth undergoing eruption with the underground bone underneath. So we'll explain what the processes are of tooth eruption from a histological point of view. The other thing we tend to look at is from the smallest level up to the organism. We do not repeat all of A and P. So, nor do we do a lot of biochemistry. Just to let you know, this is a cell. I think you remember that. It undergoes a number of processes here, including endocytosis and exocytosis. We do have cell attachments. Cells very importantly attach depending on the part of the body. We tend to look at how cells stick together. The cells that we're dealing with are what we call polarized cells. A polarized cell means the top and the apical region and the basal region are different. Even the lateral regions or basolateral regions can be different. There are special adaptions to make the cell special in the apical region, as they are the same in the basal region. And so we're going to speak about that and not go into a great deal of detail, but maybe a little bit more than you had in previous courses, things that we need for you to understand. So we'll speak about the lysosome and the peroxisomes and polyribosomes. We'll speak about the cell junctions here, whether or not they're gap junction, whether they're tight junctions, the zonular occlusions, or whether they're macula adherens. The desmosomes. Will speak about why they're important, what happens when they fall apart. Again, I just like this picture because I like it. It's good enough for me, but we do know cells come in various shapes and sizes. No, we're not going to be studying photosynthetic plants. We could, but we're not. We'll mention very briefly the nerves and whether or not we're dealing with the retinal rod cells for vision. They all have the same component, except, of course, the red blood cell, which you should remember. But we do know, if you remember, there are 4 basic types of tissues. Everything is built on these 4 basic types of tissues. There's epithelium, connective, nervous, and muscle tissue. You, in my mind, are what I call connective tissue biologists, because you work on one of the most fascinating connective tissues, and that is teeth. Teeth, of course, are connective tissues for the most part. The gingia, of course, is epithelial and connective tissue. We deal somewhat with, not much time with muscle, a lot less time with nerve, but we deal with nerve tissue, especially when it comes to pain. And pain is, and some people who know me, I always say that pain is probably one of the most important modalities you can deal with. Pain is good. Pain gives you lots of information. Some people think pain is bad, but pain tells you a lot of information, tells you whether, and it will even decide how you treat a patient. So if you remember, when you have inflammation, so if you have a patient who has pain in their mouth and they're complaining of pain, you ask what kind of pain. Is it stabbing pain? Is it throbbing pain? They mean 2 different things. The stabbing pain is when you pierce the patient's gingiva with a tool. That's the ouch thing. And then, of course, there's the throbbing pain of the pain that just never goes away because the patient may have an abscess or they may have an infection. We also know, based on the pain, that changes the pH. So when you have a pain in the oral cavity from an abscess or you have an inflammation, just remember this important thing is that the environment is very acidic. So if you have an acidic environment, what does that tell you? It tells you that, you know what, epinephrine ain't going to do that good a job. Epinephrine. It's going to tell you that lidocaine is not going to do a great job. Why? Because the local anesthetics do not work well in an acid environment. So that gives you a great deal of information. That means that before you want to treat the patient, you want to get rid of the infection. You may want to use a different type of. Have you had pharm yet? Pharmacology. Oh, you'll have lots of fun then. So you know that you have a variety of different treatments for pain. Some, when we teach you that, it's a little bit surprising what we do teach you. But the impetus for this whole thing is that when you have the inflammation, some dental treatments won't work until you resolve an underlying issue. And that really deals with the nature of what cells do. Please also understand that inflammation is very important, because a patient who has an inflammation anywhere in the body is not going to have success at dental treatment. The simple reason is that when you have an inflammation, when you have, there's a correlation with gingivitis and coronary artery disease. That we know that coronary artery disease in some cases is caused by inflammation. That we do know. We also know that patients, let's say, are diabetic and have elevated hemoglobin 1AC levels, which means their blood sugar is not under control for the past 90 days. We know that they usually have elevated chemical in the body called C-reactive protein. And C-reactive protein means that the body is inflamed, that there's an inflammation. So this is what I do know, is that there is a relationship between gingivitis and heart disease. We know that if they have an elevated C-reactive protein, they have inflammation. And elevated C-reactive protein is related to uncontrolled diabetes, getting to the good part, which means, of course, any dental work that you will do will have a high degree of failure. So as you're working in the patients and saying, oh, I've done my job, I clean out the infection, whatever, you will not resolve the issues. And whatever dental treatment has a good chance of undergoing failures. So you have to get things under control. So this is what we speak about connective tissue. So you have to think a little bit differently, for me, and that is you have to think in terms of a much more integrative approach, because you never know. We take a look at the entire patient, not just another thing. So we're going to cover everything. See, you recognize all of this. We're going to cover everything from epithelium with its different types. And that, of course, if you had me before, you know how I think about this. It gives you different degrees of protection. The thicker the epithelium, the greater degree of protection. It's to protect the body. So we can go from simple squamous and cuboidal, and we'll spend a lot of time in this, columnar to stratified squamous, stratified cuboidal, stratified columnar, transitional, and of course pseudostratified. Then of course we have the nerve. The fewest number of components we have are the nerves. Remember, what's more important in the nervous system are the glial cells, the supportive cells that are very important. This is for undergoing nerve conduction for the most part. Again, the smallest number. The largest number of nerves, of neurons in the nervous system, what we call interneurons, that are located in the brain and spinal cord, that deal with a variety of different issues, interpretation and analysis. So we want to take a good look at the nucleus, and very importantly, we want to take a look at the endoplasmic reticulum on the nerve and the rough ER around the nerve. When we take a look at the nerve cell, we see ribosomes that go around the nucleus. These ribosomes are called Nissl bodies, N-I-S-S-L-E-L. You don't have to worry about that. That's question 11 on your first exam. Again, if you know me, you know what I'm going to say. It may not be 11, 12, or 13, but that is on your exam. So you have an option. You either believe me or you don't. So if you don't, toss a coin in the air. You have a 50-50 chance that you're going to, I would say you have a 50-50 chance that you're going to be wrong. Actually, I will tell you, you have a 100% chance you're going to be wrong. So if I emphasize something's on the exam, don't believe me, get it wrong. See what I care. So now instead of with 100, you're starting with, let's say each question is worth 1.2 points. I give long exams, so you're down, let's say it's 1.5 points. So instead of starting with 100, you're starting with the 98.5. Congratulations. So we're going to take a look at that because the nasal bodies are important. The shape of the nasal bodies and the arrangement of these bodies around the nucleus can be diagnostic of various diseases. And then, of course, we have cartilage, and we have blood, connective tissue, and then, of course, we have bone. Then we have the different types of muscle, whether it's smooth muscle, whether it's cardiac, or skeletal muscle, which are striated. And then we put everything together to give us an organ. Then we deal with embryology, which studies how embryos develop. So we are interested in how you get pregnant. We are interested in what happens when the sperm meet the egg, and then the process of development that occurs after that. And I think that would be a good review for you. So we're going to do the phases of cell division. We'll do it we'll do it a little bit later on, but this is the overview. We go through the various prophase, metaphase, prometaphase, metaphase, anaphase, late telophase. So we go through all these stages in cell division and how we control them. So when we classify an embryo, there is a classification that comes out of Carnegie Mellon. These are called the Carnegie stages of human development. And we classify everything based on the number of stages that we have. It is based on what happens. So stage 1 is day 1, which is the zygote. Day 2, the next one is stage 2, which is 3 days after development. Then stage 7, 8, 9. 9 is kind of interesting. That's around day 19 to 21. And you can't see from here, we're beginning to develop the heart. The heart starts beating around day 21 to 24. And stage 10, you can see the mouth starting to develop. We'll do this in a little bit more detail. You can begin to see the process of development. What we do know is that by the time we form the limb buds in stage 21, when the limbs are growing, as you can see, you begin to see the webbing between the fingers. And really fairly early on, the embryo is fully developed. We measure development in the embryo by what we call the crown-rump ratio, the ratio from the crown to the body. We'll explain what that is in a little bit more detail. So we have really the trimesters. So the first one is what we call the proliferative period, which is 0 to 2 weeks. The next period is the embryonic period, which is really week 2 to week 8. Please remember at week 8, your face is now fully developed, your teeth have started to develop, you have all the gill slits here, and you're now considered an embryo. From 8 weeks till approximately 9 months, you're a fetus. Once you are born, you're called a baby, okay? Okay, so here we have the human embryo, 5 to 6 weeks. What you can see, this is from your textbook, and it's nice diagrams. This is not from your textbook. What you begin to see, I'm really seeing the forebrain. I see here the beginning of the nose, as you can see. I can see the palate developing here. I see the mandible developing right over here. I can see this is called the maxillary process. We're going to see all these parts growing together and then coming together to form the philtrum right over here. So that's really between 5 to 6 weeks, and you can see how early on the face has already undergone development. Then we're going to take a look at the development of the teeth. We go through the various stages. We go through the lamina phase, when we're just beginning to form, where we're going to have thickening. Please remember that as we'll review, the teeth are rather unique. The teeth are unique because they're developed from an interaction of the what we call neural crest. And neural crest is really a mixture of ectoderm and we say we have mesenchyme, which means the development of the teeth depend on the interaction of the ectoderm and the mesenchymal cells, the mesoderm. That's very important about how teeth develop. Very, very important, because what we're doing now, using this idea, we're able to grow teeth in a laboratory. So we'll spend a little time with that, but we go through the various phases, the bud, the cap, the bell, and then We really undergo morphogenesis to undergo teeth eruption. So we're going to go through that. This is from the journal Nature Genetics. I just like the picture. We use that. So that is really the goal of the course. Isn't that kind of neat? We get to do all of that in a very short period of time. So let's start here. Whoops. And we'll go on a little bit longer, and then I think we should then take a little break. But right now, let me explain the idea of my idea of a break and your idea of a break, because I think there may be two different things. My idea of a break is we get a short period of time to wake you up. I like people to move around during the break. One, I want to see if they're awake or asleep. And two, I'm a kinesthetic learner, so I like you to move around and be interactive. And I think sitting in a chair for this kind of thing is very difficult. Okay? But we're going to start this component. So where we're going to start here is, again, it is really a fundamental branch of medicine and dentistry. And again, what we study is tissues and how they relate. So I like to begin, and rather than jumping into this, I get a lot of information is that how do we get slides and whatever. So we're going to speak about that. This one is an electron microscope image. This one is of course, a brain. And we can go over why it's a brain at another point in time. But what we do deal with, microscopic anatomy, histology, and cytology. Cytology is what the cell looks like underneath, the organelles. Histology really is what it looks like, how they function in teaching. And microscopic anatomy takes the tissues and goes up and say, what do the organs look like? And let me explain this. So what histology represents for us is what tissues look like. But understand that the cells that make up the tissues are really different. And when I think about histology, I think about something called functional histology. What people think about as histology is they look at a slide and they know what it is. That makes no sense to me. The reason it doesn't make sense is that it has to be functional, because structure is related to function. So that your body structures, the histology changes depending on the stresses put on the organs. Very important in the oral cavity. Hi. Everybody got an A on the first exam already. You know that, right? Everyone got an A, right? Okay. All right. So that's what makes this course a little bit different. We start to take a look at tissues based on function. And the functionality begins to make sense. Otherwise, you're looking at a structure and saying, yeah, I see this. But what does it all mean? Why do we go through embryological development in the first place? What is the function of this? What is the stress that we have? Why is the gingiva different in different locations? Is there a functional reason for that? The answer is yes. Why does the tooth undergo this developmental process? Is there a histological reason? Yes. When the tooth erupts, okay? Humans are unique in many senses. When a tooth erupts, a cell dies. The ameloblast dies. That's the way it goes. So you don't regenerate teeth. You're not a shark. Sharks grow teeth forever. Wouldn't that be great? Animals can regenerate teeth. And I'll show you pictures that I even have a mouse and a rat regenerating teeth. You can't. Today, my ultimate goal is to, whenever I speak to periodontists, I drive them crazy because I say, because we can grow teeth, and that's really our future. Just think, the future is we're doing all the implants with dead material. But what would happen if I could grow teeth? if we could grow teeth, we call this a biotooth. If we grow teeth, you know what happens? We create living, functional teeth that respond to stress. We grow periodontal fibers. We regrow nerves. So the periodontist always asks me, or hygienist, are you putting me out of work? No, 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 I'm giving you even more work. You can even bill higher amounts. But again, the ability to do that is really 5 to 10 years in the future. And we'll take a look at that. And I'll show you some things that are going to surprise you. Okay? So histology really is the basis of pathology or pathophysiology. Very important. Okay? Embryology really comes from the Greek. It really means to swell, which is how the Greeks looked at pregnancy. They had some rather unique ideas about how embryos develop, but we are interested in the development of the individual. Just remember, all of us begin as a single cell. Okay? So this drives me crazy. We all begin as one cell. And you know mitosis. One cell divides in 2 cells, and we make 2 identical cells. That becomes 4 cells. That becomes 8 cells. We started from 1 identical cell. Now we have a little ball, which we call a morula, which we'll get to. 8 cells. And I could take a look at that and say, that's the heart. That's the liver. Right there is going to be the head. There's going to be the rear end. There are the limbs. And I can take a look at that. So something happens. So the ultimate question is, if we start out from 1 cell, what causes us to make all these changes? What causes the embryo to develop? And all of a sudden, 1 second, we have 2 cells. 1 second, we have 8 cells. And a microsecond later, when we form 8 cells, which are purely identical, I already know where the organs are going to be. What causes that? What is the fundamental stimulus to that? And believe it or not, if we knew the answer, we wouldn't be here right now, because we would know everything. But I do have the answer. And the answer is blue fairies and pink fairies. Blue fairies can cause one thing to develop in the head, and pink fairies can develop you don't like that. Okay. Can you come up with a better answer? The answer comes down to we have no clue. We don't know what makes it. And then on top of that, you and I are essentially different. You and I are almost 98% or 99% the same. Minor changes here and there that really account for differences. How does the body develop into those differences? And what also drives me crazy is that if this whole thing were easy, how do we develop disease? All right? So it's kind of an interesting process. So this is sort of like the philosophy. All right. So let's take a break here, and then we can get started. So a break in my mind is that you don't disappear for 10 hours.